separate to remove this 4 carbon to form this adenylo, adenylate. Okay, so it involves adenylosuccinate layers. Okay, so for the conversion of IMP to GMP, it also involves 2 steps. The first step is the conversion of IMP to XMP, which is xanthate. So it's catalyzed by IMP dehydrogenase. So IMP it undergo dehydrogenation to XMP. Okay, and this XMP, okay, is converted to GMP by ATP dependent reaction. So it catalyzed by XMP glutamine amidinotransferase. So this step involves the amidation, okay, amidation of XMP to GMP. Okay, it contributes the amino group. Okay, glutamine contribute amino group, so it undergo this amidation, okay, to form this GMP. If you look, okay, at this step, both steps, okay, that lead to the formation of AMP and GMP. The first route, okay, it utilizes GTP, okay, and the second route it utilizes ATP. So basically, they are not using ATP for both. So this keeps the pathway imbalance situation. Okay, one route utilize GTP, another one utilize ATP. Okay, so I hope so. Now we get our purine. So AMP and GMP. Okay. Alright, kalau ada soalan you can just interrupt. Okay. Okay, so now we go to the de novo pathway of pyrimidine nucleotide. Okay, we have 3 pyrimidine nucleotide. We have cytosine, we have thymine, and also we have uracil. Okay, uracil, cytosine, and also thymine. Okay, so in general, we know that the structure of pyrimidine, okay, saya baru tadi off video ni. You all pun tak mention. Okay, so we know that in general, pyrimidine nucleotide it has one single ring, so it has 6-membered pyrimidine ring. Okay, that made first and then attached to ribose phosphate. Okay, ribose phosphate, this PRPP. So in comparison with purine nucleotide process, is that for purine is going to be synthesized that automatically attached to this PRPP, but for pyrimidine is going to be synthesized separately, and then later is going to be attached to this PRPP. So in the process of synthesis of pyrimidine nucleotide, it requires carbamoyl phosphate. Okay, and it involves this carbamoyl phosphate synthesis too. Okay. Okay, so this is the pathway, okay, for the synthesis of pyrimidine. So it started with this aspartate. So this aspartate it reacts with this carbamoyl phosphate, okay, to form this N-carbamoyl aspartate. Okay, so this enzyme aspartate carbamylase catalyze this reaction. Okay, then once we have this N-carbamoyl aspartate, so it undergo dehydration process by the enzyme dihydroorotate to form this dihydroorotate. Okay, so this is dihydroorotate. Then this dihydroorotate is going to be undergo the oxidation process. Okay, it's going to be to be oxidized to orotate. Okay, which this reaction involves NAD plus. So this is the electron acceptor. So this dihydroorotate is converted to orotate. Okay. Okay. Next is the orotate. Okay, once we have this aspartate. Okay, so it involves the contribution of of um, aspartate in this reaction. Once it forms this orotate, we can see that we already have this six-membered ring. Okay, six-ring structure. Okay, next is going to be attached. Once the six-membered ring is formed, is going to be attached to this ribose phosphate. Okay. Okay, so at this, the first three steps, you can see the ring closure. Okay, the ring formed at this dihydroorotate. You can see this. Okay, ring closed at this deformation of dihydroorotate, and then it undergo the reaction catalyzed by dehydrogenase enzyme to form this orotate. Okay, to form this orotate. Okay, next is the now orotate. Okay, so the next step involves the acquisition of ribose 5-phosphate. Okay, so which orotate is going to react with PRPP. Okay, catalyzed by orotate phosphoribosyltransferase transferase to form this orotidylate. Okay. So now it's going to attach to this PRPP ribose 5-phosphate. Then this orotidylate it undergo decarboxylation. Okay, it undergo decarboxylation to form this orotidylate to form this orotidylate. Okay, catalyzed by orotidylate decarboxylase. Okay, so now we have the first UMP. Okay, and this UMP is going to undergo two phosphorylation steps. Okay, by enzyme kinases. Okay, which involve two ATP, meaning there are two phosphorylation process to form this UTP. Okay, this UTP. Okay, so it's a uridine 5' phosphate. So basically, so from UTP, it can be converted to CPP, okay, to cytidine, okay, by the contribution of amino group by glutamine, okay, by the action of cytidylate synthesis. So we can see that de novo pathway for pyrimidine biosynthesis is very simple. So basically, it involves three steps to form the six-membered ring, okay, which is the dihydroorotate. Okay, the hydroorotate. Then once it forms this orotate, is going to attach. Okay, the next version is going to attach it to ribose 5-phosphate. Okay, then we will get this UMP, UTP, and also CTP. Okay, so that's it for pyrimidine biosynthesis. Okay, so that's it for pyrimidine biosynthesis. So what? Okay, now you have these two. You have these two pathway. Okay, try to summarize by yourself. Try to summarize by your own understanding. Try to differentiate. Try to identify what are the donator, what are the sources for each member, okay, for each structure of the pyrimid and also pyrimidine. Okay, identify the enzymes involved so that we can discuss further in the regulation. Okay, normally for the regulation of pathway is involved the feedback mechanism, allosteric regulation of enzymes. Okay. Okay, do you have any questions? Soalan tak? Okay, so now let's proceed with the ribonucleotide. Okay, ribonucleotide are the precursors of deoxyribonucleotides. Okay, so now we have this ribonucleotide, we have this RNA. We know that we have this RNA and DNA. So to synthesis deoxyribonucleotide, we need to have this ribonucleotide. Okay, so basically, to synthesis deoxyribonucleotide, so this ribonucleotide undergo the reduction process by the enzyme ribonucleotide reductase to form this deoxyribonucleotide. Okay. Okay, so this is another simple pathway to look on the biosynthesis of thymidylate. Okay, which is this is the deoxyribonucleotide. Okay, DTMP. Okay, DTMP. So basically, from ribonucleotide, let's say CDP. Okay, how can we synthesize deoxyribonucleotide? It is the reduction because it's a reduction. This ribonucleotide undergo reduction, so then we get this deoxyribonucleotide. Okay, from this it can undergo phosphorylation. Okay, phosphorylation to form the CTP. Okay, from one phosphate to triphosphate. Okay, undergo two steps of phosphorylation. Okay, so from DCTP, it can be converted to DUTP by deamination, okay, post reaction, okay, by the enzyme deaminase. Then, from DUTP, it can undergo further reaction to form DTMP by their specific enzyme. Okay, so basically, how ribonucleotide is converted to deoxyribonucleotide is by this reduction process. So basically from CTP to UTP to TMP, so they need specific enzyme that catalyze specific reaction in their conversion from C to U or from U to T. Okay, from cytidine to uridine or to thymidine. Okay, so you need to identify, okay, what kind of enzyme, what kind of reaction involved in each reaction, in each conversion. Okay, okay, now we move to the salvage pathway. So what is salvage pathway? So basically it's a recycle, you can say it's a recycle pathway. It reutilizes the degradation product of purine and pyrimidine to to synthesize neopurine and pyrimidine. So basically the pathway is much simpler than de novo synthesis. So what is the importance? What is the importance of having the salvage pathway? So what is the importance of having this pathway? So basically, you can see that we have both de novo and we also have this salvage pathway. So you can see that how how important are these two pathways. Okay, because both pathway, both purine and pyrimidine, build up our DNA and RNA. So basically, it's a very important. So uh, by having these two different pathway helps to maintain, helps to stabilize the concentration of purine and pyrimidine bases. Okay, in order to make sure there is no problem related uh, to this leaking of, of, of this purine and pyrimidine. Okay, so the pathway of salvage, or recycle of these bases of purine and pyrimidine bases is much simpler than the de novo synthesis. So basically, it just involves two steps. Okay, the first one is it's not two steps. Okay, from adenine. Okay, the degradation product of the pathway is going to produce adenine as well. So this adenine, it can undergo the salvage pathway to form A and P, okay, by adenine phosphoribosyl transferase. So it's having PRPP. So basically, it's attached to PRPP. So by the enzyme, this APRT, it converts this adenine to AMP. Okay, and hypoxanthine. Okay, hypoxanthine can also, so basically the end product that can be utilized, the end product of degradation pathway that can be utilized by the salvage pathway basically is adenine, hypoxanthine, and guanine. Okay, so hypoxanthine also can be converted to IMP. Okay, by if you see in salvage pathway, all these three versions involve the attachment, involve the involvement, involve the contribution of PRPP to this recycled intermediate to form either AMP, IMP, and GMP. Okay, so one important enzyme that involved in the salvage pathway for purine and pyrimidine is this HGPRT, which is hypoxanthine guanine phosphoribosyl transferase. It is a very important enzyme that helps to catalyze the conversion of guanine and hypoxanthine to specific end products. Okay, why is it important? Why is it important? Okay, so because the lacking of this enzyme, can lead to this syndrome named as a Leigh-Nihan syndrome. Okay, so this Leigh-Nihan syndrome, okay, is a syndrome that led to this dysfunctioning of your neural system. Okay, when your neural system is not functioning, so it's going to reflect your behavior, okay, cognitive abilities, and also the excess of uric acid or hyperuricemia. Okay, so okay, so basically, block. Okay, so when this enzyme, HTPRT enzyme, is leaking, okay, or is not functioning, or the failure of this enzyme basically led to the cell breakdown. So basically, when you have this adenine, you have this hypoxanthine, you have this guanine. So this intermediate cannot be utilized. So it will, so the concentration will be rise, so it's going to increase the production of uric acid. Okay, so this led to this syndrome, okay, Leigh-Niven syndrome, that can you normally people that have this genetic problem will have this the neuro problem, okay, and it's going to be reflects on the behavior and cognitive abilities, and also on the excessive production of uric acid. So study has shown that this genetic problem, lacking of this enzyme, can be seen almost exclusively in male children. Okay, so it's a genetic. It's a genetic disease. Okay, lacking of enzyme. Okay, so yeah, so it can be checked through many sort of genetic tests. Okay, so this is one of the syndrome, one of the diseases related to this salvage pathway. Okay, another one is gout. Okay, I'm sure gout is quite familiar to all of you. Okay, parents, maybe our parents experience this. Okay, suffer from this gout. So we know that gout caused by excess of uric acid, and we have been told since forever, okay, we have to limit our seafood intake, okay, just to minimize the intake of food that have higher uric acid. Okay, so gout is a disease, okay, of the joints caused by the elevated concentration of uric acid in blood, in the blood and also tissues. Okay, gout can be, if we if if, if our body cannot utilize, there is no regulation, okay, regulation to convert all these end product of the degradation pathway. So the uric it cannot be utilized, and that led to the high concentration, the accumulation of uric acid. So it will led to this disease, gout disease. Okay, um, sakit juga kan? I don't know. Okay, okay. I'm sure people, anybody that experience this will listen and you can see. So you can see that it causes swollen, okay, inflamed of joints. So obviously it will affect your daily activities. Okay. So in severe condition, I know that it's going to affect, okay, your ability, especially in joints, to be practically um, okay, to be carried, your normal activity to be carried out as usual. So I am sure it will severely affect your ability when you have this problem with joints, okay, swollen, inflamed, okay. So monitor your food intake, okay, try to avoid, okay, from now, okay, whatever food that you are the one that know about your body. Okay, maybe some of you react fast with seafood, maybe some of you have no problem, but in the future you never know. So start to practice healthy diet, okay, try to avoid whatever disease, okay, because you learn, okay, you know about it, okay, the reason behind it, the pathway, the metabolism direction. So utilize whatever knowledge that you have to help yourself first and share it with your family and friends. Okay, now we know that salvage pathway utilize the degradation product of nucleotide. So now let's look at the degradation pathway. Okay, so purine nucleotides are degraded by a pathway in which they lose their phosphate through the action of 5' nucleotidase. Okay, which we know that the end product of purine nucleotide is AMP and GMP, and this AMP and GMP is converted to xanthine. Okay, and the catabolism of AMP and GMP will produce uric acid as an end product of xanthine oxidase. Okay, so AMP and GMP can undergo degradation pathway. It's going to catabolize. So the end product is uric acid. Okay. Now let's start with AMP. AMP is going to catabolize by the action of nucleotidase enzyme, and it produces adenosine. Okay, then adenosine undergo further reaction by adenosine deaminase to form inosine, then inosine that's converted to hypoxanthine by enzyme nucleosidase. If you see from this, the first three reactions, basically all reactions involve the hydrolysis of bond, okay, that catalyzed by enzyme nucleosidase. Okay, when there is the removal of amino group, so enzyme deaminase comes in. Okay, enzyme deaminase comes in, so it removes amino group. Okay, from AMP to adenosine, then adenosine to inosine, then inosine to hypoxanthine. Okay, so the third step involves the removal of ribose group to form hypoxanthine. Okay, and hypoxanthine undergo further reaction by xanthine oxidase to form xanthine. Okay, to form xanthine. Then the second reaction involves xanthine oxidase. It led to the formation of uric acid. Okay, so basically reaction that catalyzed by xanthine oxidase, okay, it removes hydrogen peroxide. So this is hydrogen peroxide. Okay, and GMP. Before GMP, it utilized enzymes nucleotides as well. So from GMP, it it converted to guanosine. Then guanosine, it undergo the removal of ribose by enzyme nucleosidase, and now we have this guanine. So guanine, okay, both fraction will meet at the will produce xanthine, okay, before it can be converted to uric acid. Okay, so guanine is going to be converted to xanthine by the action of guanine deaminase. Okay, so guanine, there's a removal of amino group here to form xanthine. So it removes this amino group, okay, to form xanthine. Okay, so basically catabolism of purine nucleotides involve the formation of uric acid. Okay, so 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 people that have problem with the high concentration of uric acid, it may have problem with the metabolism reaction. It may be enzymes, maybe maybe certain enzymes not functioning, especially xanthine oxidase, right? Okay, so okay, so this one is catabolism of pyrimidine nucleotides. Okay, pyrimidine nucleotides. So let's say we look at the thymine. Okay, thymine, it will undergo the first step, okay, by enzyme dehydrogenase to form dihydrothymine. Then the hydrothymine undergo the reaction catalyzed by dihydropyrimidinase okay, to produce this uridoisobutyrate. Okay, then it undergoes further reactions until it produces methyl malonyl semialdehyde. Methyl malonyl semialdehyde. And this is methyl malonyl semialdehyde. Okay, so this end product of the pyrimidine nucleotide is further degraded to succinyl CoA. Okay, so succinyl CoA is the intermediate of TCA cycle. So later it can enter TCA cycle for further reactions. Okay, so this is summary. Before we go to summary, do you have any questions? Ada soalan tak?
Doktor,
Ya.
nak tanya.
Mhm.
Untuk purine tu kan ada GMP dengan AMP tu kan.
Okay.
Itu apa kaitan dia dengan purine?
AMP. Okay. AMP stand for?
Tak ingat sekejap.
What is AMP?
Adenosine.
And what is purine? Okay, adenosine monophosphate. Okay. Let's go back to AMP. Okay, so this is your AMP, adenosine 10. Okay, so okay. Soalan first saya tanya tadi kan, what are purines? Okay, what are purines? So what are purines tadi? We have this ANG apa? Adenine and
Guanine.
guanine. Adenine and guanine. Okay. Okay. Tengok dekat sini ya. Okay. Purine. Okay. Kalau in nucleotides, it's known as adenine and guanine. But when it is in the let's say tengokkan nucleoside. Okay. It is known as adenosine. Same goes for guanine, it is going to known as guanosine. So macam AMP, GMP, you know that AMP is adenosine monophosphate. So basically, purine nucleotide to form this adenine, basically it will function as adenosine monophosphate as AMP and also GMP. So AMP basically is adenine, but adenine. Okay, we know adenine is only purine kan? Adenine purine kan? Okay, adenine is purine, but we would like to have why. Okay. So this adenine, so AMP basically is something that you have attached it to ribose. Betul tak? Monophosphate. Okay, look at this structure. Nampak tak? So you have this is your adenine, but when it attached to ribose, when it attached to phosphate, it's going to form this adenosine monophosphate. You look at the idea of it. The difference between bases, adenine is a basis, it's a purine basis. So nucleotide itself, it need to have that three components. Kan, betul tak? Okay, nucleotide itself, it has to have these three components. Purine bases, ribose sugar, and also phosphate. Okay, so adenine is a basis. It's a basis only. So once we synthesize the purine, okay, the first time kalau purine tadi, the first few steps tu we synthesize the first 5-membered ring, then followed by 6 ring. So for purine, dia automatically memang it synthesized attached directly to ribose. Kalau compared to pyrimidine, we need to synthesize the 6-membered ring first before we attach to sugar, to ribose and phosphate. So basically, this is your purine. This is your adenine, for example. This is your adenine. But when it attached to ribose and phosphate, we form nucleotide. Okay, that known as adenylate or adenosine monophosphate. So this is your, okay, when we talk about de novo synthesis of purine. So purine ni sebenarnya we want to synthesize this purine. But the importance of purine, because we want to have this nucleotide. Nucleotide need to have these 3 components. Okay, Yazwani, are you okay?
Okay, faham dekat?
Okay, do you get the idea?
Ya.
Alright, same goes to GMP. Okay, sama je. Okay, so the process basically, the synthesis phosphate involves the formation of purine. But to function as a nucleotide, you need to have that. That's why dalam purine dia terus je. Okay, that purine structure terus attached kepada ribose. Okay, dia terus je. Okay, dia terus je dekat ribose kat sini. Okay, dia terus sebenarnya. Okay, compared to pyrimidine. Okay, any other questions?
Lepas tu doktor nak tanya lagi.
Okay.
Untuk pyrimidine tu kan last step dia kan dia akan jadi CTP kan?
Pyrimidine. Okay, then?
Kiranya CTP tu pun macam tak ada juga ke? Macam purine juga ke?
Sama. Okay. So for pyrimidine tu sama. Okay, pyrimidine we have thymine, uracil and cytosine. Betul kan? Betul?
Betul.
Okay, alright. Okay, but to function as a nucleotide, you need to have that 3 component. Sama je macam purine. So that's why dalam biosynthesis of pyrimidine, okay, first is it started to synthesize to build up the 6-membered ring. Okay, it started to synthesize the 6-membered ring. Then it needs to attach to ribose phosphate juga. Sama. So it require this another 2 components. Okay, apart from bases, apart from pyrimidine, it need to be attached to ribose sugar and also phosphate, so that it can function as a nucleotide. Okay, so and also if you notice both pathway, there must be phosphate and ribose sugar. But once it attached, okay, once it's produced as a UTP, so basically UTP is uridine triphosphate. So basically it refers to uracil. Okay, uracil. But from uracil it can be converted to CTP as well. Okay, CTP for cytosine. So similar. Okay, similar principle. Okay, sama je teori dia. In theory both. Okay, to function it needs to have that 3 component, complete component of phosphate, ribose and also nitrogen bases. But because for ribose sugar we don't have any, there is a basic intermediate. Okay, it's a basic sugar. Okay, ribose phosphate. Maybe we can have it from PPP. Okay, ribose 5-phosphate. So maybe from ribose undergo phosphorylation to have ribose 5-phosphate. Okay, but if you can see that both pathway utilize PRPP, which comes from ribose 5-phosphate. Okay, so sama. So basically UTP, CTP, it refer to you punya bases which are uracil, cytosine and also thymine. Okay, mungkin you tengok nama dia macam lain sikit, but basically it refers to, because why is the name is because it has other component, okay, dalamnya. Okay, UPGH, okay, refers to uridine monophosphate. Okay, tak adalah you tulis uracil monophosphate, no. Okay, because uridine, it shows that it has this amino group, it has undergo phosphorylation, okay, it has so many things. Okay, that name basically it reflects, okay, whatever the process that it has undergo. Okay, it went through, okay, there are few versions, okay, carboxylation lah, decarboxylation, many things. So it reflects, okay, whatever version that has been provided, okay, to form this nucleotide. Okay, so basically in the system, our purine, our pyrimidine present as AMP, our purine and pyrimidine present as AMP, present as GMP, CTP, UTP, okay, macam tu. Alright, okay. Okay, ada soalan lagi kah? Before we summarize this, summarize sama lah. Yes, yes, okay. Again, tak faham. Contribute phosphate group. Okay, okay. Bukan, okay, okay. Kan the first soalan quiz yang saya tanya, the first tadi mula-mula tu is, what is the component of nucleotide? Okay, you need to have this phosphate. Okay, you need to have phosphate, you need to have the soluble sugar, and you need to have bases. Okay, okay, this is pyrimidine bases, right? Okay, now in this pathway, we would like to synthesize. We look, okay, specifically on the synthesis of pyrimidine, for example. Pyrimidine kan? Okay, but kita tak nak, we don't want pyrimidine bases, okay, sahaja. Okay, we would like to have one complete nucleotide. Okay, kita tahu kan, okay, kalau purine we have adenine, we have guanine. Okay, kalau untuk pyrimidine we have cytosine, we have thymine, we have uracil. Okay, kita nak tiga ni, thymine, uracil, thymidine, thymine. Okay, cytosine, thymine, uracil. We want these three, but these three bases kita nak, we want to have this nucleotide that have that three bases. Okay, and that three bases dalam nucleotide it present as, kalau uracil as UMP ataupun UTP. Okay, when we have cytosine, okay, in a nucleotide, okay, dia akan present as CTP. Contoh, cytidine. Okay. Kalau thymine, dia akan present as a TMP, thymidine monophosphate. Okay, it's a process of synthesize you punya thymine, uracil semua tu, tetapi we don't want that because we want nucleotide. Nucleotide need to have these three, dia perlu ada komponen. Yang tadi tu kita synthesize ni je. Okay, yang banyak-banyak tu kita synthesize ni. Okay, but it to be function as nucleotide. It tak contribute pun, but it need to be attached to ribose sugar, to phosphate. Okay, and dia bolehlah function as nucleotide. Okay, you all faham ke tak? Nanti saya akan share cara lain apa, explain.
Doktor, kiranya
Okay.
kiranya kan,
Okay, kiranya apa?
CTP tu dia dah satu nucleotide yang complete lah maksudnya.
Oh yes. Okay, now this is your nucleotide. Basic. Kan? Okay, we have phosphate, we have ribose sugar, and we have nitrogen bases. These nitrogen bases either pyrimidine or purine. Okay, so this two pathways yang kita discuss tadi is to synthesize pyrimidine ring and purine ring. But both process basically, bila dia, okay, kalau contoh purine, when it synthesize purine ni, dia terus attach kepada ribose and phosphate. So dia dah jadi satu nucleotide. We want to have this nucleotide to function that have a very important function in our daily life. Kita banyak, kita punya DNA storage semua kan, kita punya genetic information, semua in nucleotide. So we would like to have this nucleotide. But dalam pathway tadi, we discuss on the synthesis of this purine. Purine, walaupun we discuss on the synthesis of purine, tapi dia automatically attached to these two, ribose and also phosphate, because we want to have this as nucleotide. Okay, contoh macam adenine. Okay, adenine, once it attached to ribose and phosphate, it known as a AMP, adenosine monophosphate. Kalau dekat sini guanine, okay, bila dia attach pada ribose and phosphate, it known as GMP, guanidine monophosphate. Same goes kalau pyrimidine. Kalau dekat sini bases ni adalah uracil, it is known as UTP. If this basis known as if this basis is cytosine, it is known as CTP. Faham tak? Okay. You all confused sebab nama dia triphosphate ke? That's why if you notice in this in the formation of, let's say, kan purine kan, we synthesize this purine, but it automatically attached to ribose and phosphate because we want this nucleotide as a whole. Okay, and then okay, as a whole, then it's going to form this AMP and GMP. So this is adenine bases. Okay, so we know as adenylate. Okay, when it has ribose and phosphate, okay, so it is known as adenylate. Kalau guanine bases, okay, guanine base kat sini, okay, that attached to ribose and phosphate, it is known as guanylate. Alright. Monophosphate ke triphosphate ke, it based on dia punya phosphorylation reaction. So basically this is nucleotide. Okay, this is your nucleotide, AMP, GMP, CTP, CDP. Okay, so that's why if you have textbook kan, if you have textbook or you go through any online sources, okay, you will find this system. Okay. AMP, GMP, CTP, CDP tu, okay, because dalam DNA it is known as AMP, GMP. Alright. Ada yang nak clarify lagi?
Doktor,
Ya.
ni lepas tu kan ada dua pathway kan? Itu dia jadi serentak ke dia macam take place juga?
Okay, the second pathway, okay, we have de novo pathway, meaning is newly synthesized. Okay, newly synthesized from intermediate. Okay, we have PRPP, we have glycine, we have aspartate kan. When we have that intermediate, we can synthesize new. Okay, it's a new from scratch. Okay, another one is salvage. Salvage pathway it recycle. Okay, so last topic tadi we discuss on the degradation pathway. Degradation pathway is going, there will be removal of adenine, of guanine, and also hypoxanthine. So these three components is going to be recycled. It's going to be recycled. It's going to be used to produce AMP, GMP, and IMP. So basically, uh, in general, I think it's going to the pathway. Basically, in general, I think yeah, it's going uh, to produce nak cakap serentak tu tidak lah, but as long as we have once the salvage pathway uh, occurs, is going to be cycled, because the importance of the salvage pathway is it helps to balance. Okay, we don't want any uric acid formation that increased. Okay, so the salvage pathway help to utilize all these adenine, guanine, hypoxanthine, and helps to recycle and reconverted this intermediate into IMP, GMP. Semua itulah. So it helps to decrease, to maintain the uric acid level. So saya rasa it can happen spontaneous. Okay, just to make sure this is a part of one important thing is to maintain our nucleotide level, and also to help to balance the uric acid concentration. Okay, macam kita tengok tadi Lena, Helm and Gao, when we have high concentration of uric acid, so we know that one of it is the enzyme that is very important in salvage pathway, which is HGPRT. Okay, hypoxanthine guanine apa tadi? Okay, so it can happen at the same time, probably. I don't know. Okay, I don't really know, but you need to know the importance of the salvage pathway. Okay. Nanti saya check dia serentak atau tidak. Okay, but I think as long as there is an degraded product, I think it's going to be converted directly to IMP, GMP. Okay, it's going to be recycled. Otherwise, let's say for example, Lena-Hansen syndrome, bila that enzyme is not functioning, so the intermediate product is not going to be recycled. So there is a excessive production of uric acid. So kita tak nak macam tu. Okay, kita tak nak the high the rise of uric acid concentration. Okay. Sebab bila ada uric acid tu memang sakit kau apa. Okay. And in fact, for Lena-Hansen syndrome, you have problem with your neuro system. Okay, you punya heart metabolism, you punya behavior, your uric acid concentration is also high, so we don't want that. So that's why salvage pathway is very important. You can see that the salvage pathway dia punya reaction is a very simple one step reaction. Okay, from adenine to AMP straight away. Okay, from hypoxanthine to SMP straight away. So it make the process, it keeps the process simple as they can. So to make sure there is no accumulation of end product that can lead to increased concentration of uric acid. Okay. Okay guys, has any?
Okay, doktor.
Okay, summary. This one is a basic summary. Okay, that I stated here. Okay, so nucleotide is even though it's the first sentence is very important. You need to understand the picture of nucleotide. So you need to have this nitrogenous bases, pentose sugar, and a phosphate group. Okay, so nucleic acids are polymers of nucleotides joined together by phosphorus. Okay, so engages okay, between 5' hydroxyl group of one pentose and the 3' hydroxyl group of the next. So the second statement, it explains about the connection, the double helix, okay, how one nucleic acid linked to another that form double helix structure. Okay, so there are two types of nucleic acid, DNA and RNA. So the nucleotides in RNA contain ribose and common pyrimidine bases are uracil and cytosine. Okay, so in DNA, the nucleotide contains 2' deoxyribose and the common pyrimidine bases are thymine and cytosine. Okay. Okay, this one yang saya highlight tadi lah. Okay, the purine bases are assembled already attached to the ribosome. Okay, in the de novo synthesis of purine, kita synthesize dia terus attach pada ribosome. Okay, alternatively, purine bases released by the hydrolytic degradation of nucleic acids and nucleotides can be salvaged and recycled. It will be reutilized, it will be recycled and converted back to AMP and GMP. Okay, so purine salvage pathway are especially noted for the NAD that they say, okay, and the reclaimable effects, they are absent. Okay, so the end product of both purine and pyrimidine degradations is uric acid. So we need to make sure that the end product is reutilized, so there will be no accumulation of these bases, so that can lead to the high concentration of uric acid. Okay, so few purines can be salvaged and rebuilt into nucleotides, and genetic deficiencies in certain salvage enzymes cause serious disorders such as Linnaeus syndrome and ADA deficiency. ADA deficiency ni saya tak ingat lah apa nama dia. Okay, saya check balik. Okay, so accumulation of uric acid crystals in the joints possibly caused by another genetic deficiency results in gout. Okay, so enzymes, okay, enzymes is very important. Okay, so it's a target, okay, targets for any treatment, okay, any diseases. Now people are working with enzymes, okay, to understand the characteristic, behavior, the reaction of enzyme to with the hope of overcome any to build up, to suggest new treatment for certain diseases. Okay. Okay, now we complete, okay, we have completed this topic. I hope you able to, this is the LO lah. I hope you able to describe, the important one is able to describe the de novo and salvage pathway. I hope you be able to distinguish and draw the nucleotide structure, and you understand the importance of nucleotide, what are the role of the nucleotides, and also understand the catabolism process of purine and pyrimidine. Okay, inshallah, kalau if you keep revision, revise, it's not that difficult. Okay, me myself used to be undergrad student, sama je macam you all. Okay, and what I love about biochemistry is kita ada pathway. You don't have to remember, but I think the key word is you have to understand. Okay, whatever reaction involved, enzyme apa, dekat mana, okay, replacement here and there, contribution with amino group dekat mana. Once you understand that, it is easier for you to remember. Okay, so that's the most important thing. Okay. Okay, now we have another few minutes. Okay, saya sebenarnya nak discuss pasal regulation. Okay, regulation of this biosynthesis of purine pyrimidine. Okay, in general, okay, let's say, okay, saya tak ada slides on regulation. I'm prefer to use this slide to explain. Okay, let's say, okay, we know that the key intermediate, okay, the key intermediate in both pathway is 5-phosphoradicyl pyrophosphate, PRPP. Okay, remember that, the key intermediate. Okay, we know the key intermediate. Okay, so basically we can say that this both pathway is precisely regulated events. Okay, is a regulated events and coordinated by feedback mechanism. Okay, biasanya in regulation of biosynthesis of pathway, there are two regulation system. Either one is feedback mechanism and one is another one is allosteric regulation. Okay, why we need this regulation of biosynthesis process? Okay, why is it important to understand the regulation of pathway? Okay, because in general, we want, okay, the regulation is very important to ensure their production. To ensure the production of whatever intermediate. Okay, for example, specifically you want to have this nucleotide to be produced in appropriate quantity. Okay, we don't want any excessive production of uric acid. We don't want to have any problem, any irritation to produce DNA. So we need to have this regulation. Okay. So basically we can say that, okay. So now this is your PRPP. So by having this pathway, okay, we know that PRPP is a very important intermediate in the purine and pyrimidine biosynthesis. Okay. 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 Now, okay. We have discussed on the diseases related to purine, purine. But for pyrimidine metabolism is a very is quite rare disease that related to pyrimidine metabolism. Okay, uh, because the products of pyrimidine metabolism is highly soluble. It's soluble in water, so it can be transferred, it can be converted much easier compared to purine. Okay. Okay. So in general, one thing information that you need to know, IMP, inosine monophosphate, is the first. Okay, this IMP, inosine. Okay, inosine monophosphate is the I can say that parent nucleotide, okay, of purine, which from IMP it can form, it can lead to two different routes to form AMP and GMP. IMP, okay, from IMP it led to formation of AMP and also GMP. Okay, okay, so you need to know that. Okay, so in general, for purine, for the biosynthesis of purine, it depends on the PRPP pool sizes. So the PRPP pool sizes regulate, okay, the purine nucleotide synthesis. Okay, because it's going to reflect the utilization, the synthesis, and the degradation of purine nucleotides. Okay, this the pool sizes of PRPP. Okay, number one. Okay, so we know that PRPP is synthesized from ribose biphosphate by PRPP synthetase. Betul tak? So dekat sini, so we have this ribose biphosphate kat sini that synthesize PRPP by the enzyme PRPP synthetase. So this enzyme PRPP synthetase is sensitive to both AMP, GMP. So that one is the key, the how it regulates. Okay, if they sense that AMP and GMP is in high concentration, maybe it's going to stop. Okay, the activity of PRP3 synthesis. They're going to produce any PRPP. It's not going to convert, okay, PRPP yet. Okay, so here we can say that AMP and GMP is the allosteric regulator for the PRPP synthesis. Okay, number one, PRPP synthesis. So AMP and GMP is an allosteric regulator for this PRPP. Okay, then, okay, this is, okay, saya mention tadi kan, the first step is the initial committed step. Okay, initial committed step. Kat sini berlakunya apa? The displacement of pyrophosphate with amino group. So enzim dekat sini adalah PRPP glutamyl amidinotransferase. Okay, PRPP glutamyl amidinotransferase. Okay, so so this enzyme, PRPP glutamyl amidinotransferase, is inhibited by AMP GMP. So it is feedback inhibited. Okay, feedback inhibited by AMP GMP. So kalau if at the moment we have high concentration of AMP and GMP, so this enzyme is going to be inhibited. So there will be no conversion of PRPP to PRA. Okay, so this is the first committed step. Okay, dalam purine nucleotide synthesis. Okay, another one. Okay, so we know that there are two enzymes that regulated, okay, by AMP and GMP. Another one is this aminosuccinate. Okay, AMP also inhibits the enzyme adenylosuccinate synthesis. Okay, AMP inhibits the enzyme of aminosuccinate. So, bila ada high concentration of AMP, it automatically inhibit this enzyme. So, it's not going to convert AMP to aminosuccinate. Okay. Okay, same goes dekat sini juga lah. GMP dia akan inhibit IMP dehydrogenase. Okay, so these are this is how regulation in purine biosynthesis occurs. Satu dekat enzyme PRPP synthesis, and second one is dekat the first step tadi, and followed by aminosuccinate synthesis and also IMP dehydrogenase. Okay, both all enzymes is going to be inhibited by AMP and GMP. So basically, these two end product is very important. Okay, because it helps to regulate the purine nucleotide biosynthesis. Okay, okay, ada masalah? Take note. Okay, tak ada slide kat tu. Alright. Maksudnya simple topik tu saya tak buat slide. Okay, we have to take note on that. Okay. Okay, kalau pyrimidine biosynthesis, this is saya kena laju. Okay. So pyrimidine biosynthesis, so we know that carbamoyl mana? Carbamoyl synthase. Okay, this enzyme, carbamoyl. Okay, so this aspartate transcarbamylase is inhibited by CTP. Okay, by CTP. So kalau there is high CTP, it's going to inhibit this reaction. So it stops the conversion of aspartate to carbamyl aspartate. So dekat situ je lah untuk pyrimidine. Okay, so this is the allosteric regulation for pyrimidine biosynthesis. Okay, so kalau untuk ribonucleotide, obviously regulation dia dekat ribonucleotide juga this enzyme is going to be regulated. Okay, Okay. Any questions? Ada soalan tak?
No, Doctor.
Okay, I hope we have covered on the introduction part on the de novo and salvage pathway and also degradation, and just now on the regulation of purine and pyrimidine biosynthesis. So I hope you get something from today's lecture. So if you have any questions, you can direct text me. Anyone please, kalau saya tak reply in few hours, please remind me again. Kadang-kadang saya memang terlepas pandang. Okay, so I have now obviously dengan PDPR, dengan work from home, I have so many. Okay, sekolah pun saya tak tahu lah berapa banyak setiap subjek and everything. So kadang-kadang saya terlepas pandang. Okay, kadang-kadang dah ke bawah dari organisasi. Okay, just remind me again. Okay. Alright. Okay, if you don't have any questions, it's already 11 o'clock, right? Give it like 5 minutes lah. Ada soalan boleh tanya. Kalau tak ada, I'll see you next
Doctor?
Wednesday. Yes.
Do we have class on Wednesday? Because it is a public holiday, right?
Is it? Dia apa?
West Saturday.
Okay. Okay, tak apalah macam tu. Okay. Okay. Can I just give you one simple tutorial for Wednesday, so you can just do it anytime that you want. But you need to submit your
The report.
yes.